Wonderful. Good morning, everybody, again. So I know that a lot of the participants that are attending online were not connected yesterday, so please allow me for a short introduction about the foundation, just for a minute. So I'm Francesca Virlis, Managing Director of Reef Renewal Foundation Bonaire. Reef Renewal was founded in 2012 with the goal of assisting natural recovery of coral reef using active restoration. We use fragmentation and coral breeding as techniques to propagate thousands of corals or young individuals, let's say, that are planted back to the reef. We currently have a nursery capacity of over 23,000 corals planted back to the reef, over 60,000 corals. And all this we did it with the help of the community. We engage local and tourist divers that get trained through the dive shop, our dive shop members. They volunteer for us and allow us to do what we do. But how did we get there? Well, it happened quite fast, I have to say, encouraged by the positive result that we were witnessing, in our nursery and the outplantings. We were seeing like corals growing healthy. So we started increasing capacity in the nursery and producing more corals and outplanting more corals. And then we started with let's say a dozen trees, a couple of hundred corals in the nursery, only 2 species. In just a few years, we were producing thousands of corals, right? Every year. First of all, not necessarily the team grew exponentially as the corals. Actually, for a very long time, we were just 2 people. But also, other than that, I felt like we didn't have really a good grasp of what we were doing, right? We were just producing more and outplanting more, but okay, I knew that we were doing well, but how well? Also, what we were doing wrong, and there were maybe some sort of inefficiencies, let's say, that maybe I could have worked on, because it's too easy to just say I need more stuff, right? But maybe that's not the solution, right? Maybe you need something else. Also, because if you increase your capacity, you're nurturing, you're installing more trees, but you don't use them effectively, actually you're just wasting time. You're just wasting time maintaining these trees, but this doesn't translate in actually outplanted corals. So, to answer these questions, first, I had to think, okay, what I need to know. And that's the most important thing, because the tendency normally is to go collect data. And I remember at the beginning, I spent so much time measuring corals, 2 millimeters per month, 2 centimeters per month, and was very much useless in terms of production, right? So I really needed to know, first of all, as soon as we start scaling up, I wanted to know, when you apply, for example, for funding, right? What are your goals, right? And how are you be able to achieve it in how long? For example, you have 500 square meters of reef that you want to restore, or 1,000 square meters, or 2,000. How many corals do you need? How long it takes you to produce those corals? Which species are you going to start with? Which will be the second species on the second year? And the bigger the project becomes, the more important are these questions, right? And to have a very well-defined strategy ahead of time, because when you ask for money, you need to have very measurable objectives, but you also need to make sure that you can deliver it without actually risking too much. Because maybe the first project could be good, even if your results are not that good, let's say, but then you need to ask money again, and then you start losing credibility, right? And so for me, project planning, have enough information to properly plan was one of the most important things to know. I wanted to know what I could do better, right? So to improve our inefficiency, right? I wanted to know where we were wrong, but not at the end of the project. Right? When corals were dying and maybe the reef wasn't restored. That was too late. And not at the end of a research project when the paper was published after 4 years. I needed to know in the first month right away so I could adapt or change something along the process. And all that because we wanted everything here to translate in more coral alive. So how did he do that? For a second, we had to set aside our marine biologist mindset, not think as a reef ecologist anymore, but as farmers, right? If you go and look at a manufacturing process, right, we had to kind of tap into that world and think in a different way. I think about indicators or performance indicator as production volume, as production time, how long it takes me to produce this amount of corals, scrap rate. Sorry for the corals, I don't call it scrap, but if they die, they're useless. It's like you're losing productivity, right? So where I might be able to produce on time. Also, what are my costs, right? So also downtime. Are there moments in my process where we're actually not producing? All these questions, all these terms and indicator, what I have to do is just translate. This is kind of almost common sense in any manufacturing process, but not so common in our world. So what I had to do is just translate it to the coral lingo, right? So the beginning probably is familiar to all of you, because what you need to do initially is to establish a tracking system underwater. And this is how we did it. This is a nursery unit that in this specific case is a tree, but we call it unit because it could be a rope or a tray in our database, right? So multiple units constitutes or make up for a nursery. So in our database, each unit, and we have over 260 units now underwater. In our database, each unit is associated. You see, for example, in this code, you have a site with the GPS coordinate of where that unit is installed, so the GPS coordinate of the nursery. Then you have the position, the specific position of that unit in the nursery. At Climb Bonaire, we have 60 trees in that nursery. Where is this tree? I needed to know. Is it the second, the 25th, the 35th position? And that we call it location. And this is this second part of the code. And then associated to this code, we know when the tree or unit has been installed and removed. It was important to know for us how long has that tree or unit been underwater, right? Or is still there actually, or we have an empty location to be filled with another unit. And of course, for every design, you have a different coral capacity. So in our database is associated to that unit, the corals that you can actually put on that unit. And then we talk a second code that is coral related, the coral ID, because in a unit you can put different species, different genotype, but the unit stays there, right? So we associate to each unit a coral ID. The coral ID in our database, and that's the code that you see on top, refers to the species, the genotype, and in some nurseries, for example, but it's optional, you can also specify if it's a broodstock or parent colony, so will stay in the tree, or actually those corals are meant to go and be outplanted. In particular for the boulder corals, we do this. And of course, for that specific genotype in our database is also, that information is linked to where we originally are harvested, the date, the site, if there are some pictures, and the depth. So once you set your basic, then the most important thing, which information you want to collect, right? Which data? Sounds easy, but actually this is probably the most important question, because at the beginning you tend to collect a lot, but I can't stress enough, keep it simple. I don't need to organize 1 week of monitoring to monitor all the nursery and outplanting site once a year. That's not useful for me. I wanted to know at any given time how the nursery are producing, how our planting sites are. So that module of like once a year, once every 6 months didn't work for us, right? If corals suddenly die tomorrow in the nursery, I want to know within a week, right? So every time we work in the nursery, we want to record some set of basic information, and shouldn't take more than 2-3 minutes. Otherwise, you're not doing restoration. You're just collecting data all the time. So in 60-minute dive, if you work on a tree, like I said, recording to your slate what you did shouldn't take more than 2-3 minutes. Again, this is what worked for us, right? Not necessarily for everybody. It depends on your goal and your priorities, going back to what I was saying before. So of course we record some basic metadata, who's collecting information, date, temperature. Of course, you need to tell on which unit you're working and which species and genotype, just to make sure that the database is correct, that the association unit and coral ID is correct, so you record both tags. And then you start collecting your data. So we simply record how many corals we add that day, right? Adding means fragging. You frag more corals, you put it on a tree, so you are occupying some available spots on that unit, right? Fragging or just transferring from another unit, right? So you need to record that information. Same, you record if you remove and make more available spot on the unit. You also need to record why you remove them. Maybe we're just outplanted, maybe transferred to another unit, or maybe they died, and that's why you remove it. And if those corals were dead, you also need to write down if it was a disease, predator, competition, the reason why they died, right? Again, also make it simple. We also train the staff. This is normally information that the staff or the team leader will record, not just maybe a basic volunteer, right? Entry-level training. Another very important information is every time you go, give an estimate on that unit of how many corals are ready to be outplanted. You know that the capacity is 100 coral. I don't need to know if it's 71 or 72. I don't care. Tell me, like more or less 80, 20, 30, 50, the order of 10. I want to know how corals are growing there. These are the basic information. And then let's see how we use those. So we elaborated a database thanks to an angel volunteer that is an expert in elaborating database and knew exactly what we're doing and helped us in a process that lasted several years, actually. In automatic, everything, all these reports are generated. First, production volume. Like I said, going back to the question, I want to know how many corals I produce, every unit produced. So coral produced means broodstock, so the initial parent colony that you put on that tree, and the corals that were added on that tree or unit. Production time and ready to outplant time are indirect measurement of growth, because again, 2.5 centimeter, 3 centimeter didn't help me to understand where we were in our nursery. But if I know how many days since I put the first coral on the tree till the time that tree or unit was full of corals, that's how also an indirect measurement, accurate enough, right, for us, of growth. Because if the broodstock don't grow, you cannot cut them to fill the tree. But a tree could be full, or unit, sorry, I give the tree because that was the sample here. A tree could be full, and the corals would be all small, or full and the corals be all ready to be outplanted. That's a completely different time point, right? So we needed another metric, and that's the ready to outplant time. The time since the tree is full for the corals to be, for 80% of the corals to be ready to be outplanted, right? And I will give you some example. I know this, bear with me with this number, but that's just to say how we measure how fast coral grow in terms of days. Also, an important information, how much of the corals that you produce actually turn into outplanted corals. This is very important, how efficient you are, because you can produce a lot of corals, but if you don't outplant them at the same ratio or a specific rate that is your goal, let's say, then it's also not good. So that was production volume. And then we spoke about scrap rate, right? Failed units, coral that died along the process. Again, automatically generated. If in our database, I click on a unit, on a tree, on a nursery, then I know how many corals died there. And you're familiar with mortality, right? How many dead corals versus the total amount of corals produced in that unit. And then also, because we record, right? Remember the data that we record, and we say if you remove a coral, you need to know why. Then the database can tell you as well how many corals died because of disease in that unit, or because of predator or predation or competition, versus the total amount of corals that died on that specific unit. And that's the unit that failed, right? The one that didn't go on the reef. Also downtime. If you need to remove a unit out of the water, that's downtime, unless you immediately swap it for another one and you're still feeling core. That means that there is an empty location on the reef that you're not using. Also, downtime is kind of, like I said, measure your inactiveness. If you leave a tree, for example, if your tree is half full and the coral is getting big, but you don't go there and frag them, and you don't reach your full production, right? Like you don't cut your corals to fill the tree, then you don't reach your production time. So actually, those coral that you think are ready to be outplanted, in reality is not. In reality, they are ready to be fragged to fill that tree, right? So leaving also empty spot on a tree is inactiveness. For example, you outplant the corals, but you don't go back to refill the tree, is inactiveness again. So occupation is a very important metric for us as well. So how many spots available you have on that unit? Don't panic. I'm not going to go through all this data. I just want to point out a few important information. And this is kind of a standard report for every nursery and for every unit that we get an automatic from our database. So what you do, you select your nursery, and you see here is Knife Nursery, small nursery at Bonaire. In the first column, you see all the units there. Those are trees and all the genotypes and species that are installed on each unit. This is actually a cycle of production so when a cycle of production, the corals go out, right? We close the cycle. It stays in the memory database. You can open a new one. So this was a cycle closed, for example, last year. So I can see how many spots are available, but very important information. I want you to take a look at the production time. So we started with the same amount of coral on these two trees, approximately 8 or 11 corals, almost the same, right? It doesn't really make a difference. And then for this tree, we reached the production time. So we were able to fill that tree in 435 days. It's a little bit over a year, right? For this genotype, so same species, same location, the trees are next to each other. To fill the tree, it took us 649 days, which is 210 about days of difference. It's 7 months. So to fill a tree starting from the same amount of corals, it took us for these two different genotypes, 7 months of difference. And you can imagine if you have a 2-year program, like if I use only this genotype, I would never be able to get these corals outplanted on time. But if I use this one, maybe this is just an example. And you might think, oh, okay, maybe it took you longer because you lost a lot of corals, a lot of scrap or failed units, right? But then you look at survivorship, and this tree was 97%, this was 100%, definitely not a significant difference. There was not a high mortality on the second tree. So the only explanation that these coral were growing slower. Nothing bad with it, but you need to be aware. Maybe it's good to go at previous cycle of production for that genotype and see if it was always like that. Maybe something happened specifically, right? But the fact that at that nursery, the others were growing fast made me think that it's really genotype related, because all the other genotype were growing fast. So what you can do then still, it's another automatic report that we get is like, let's see where that genotype is replicated also elsewhere and see if it's happening same also in other places. Let's see, this should be a video. Yeah. Oh, with the mouse. Okay, thanks. This one? Which this one or the pad? Yeah. Sorry. I don't see the. Let me see. Yeah. No, that's went to the wrong. There should be a video here. Click. I think you should press it. Yeah, that's. I'm clicking. Maybe the space bar? No. Let's see, it takes a second for the video to start anyway. I recorded like a couple of seconds. So.
Sorry, no, I don't.
Yeah, it's okay. It's okay, I'll go through it. No problem. Let me try one more. Well, the video showed very simply anyway, that in our database, you can just click on one of the species there and it will open up, of course. And then for that species, you see all the genotypes and how your full capacity, over the full capacity, let's say the genotype, you allocated, let's say 20% of your total capacity to that genotype, or 10% or 5%. So the percentage of allocation for that specific genotype. And that's important because if you know that the genotype is very well performing, you want to dedicate a little bit more space to that genotype versus other, maybe. That could be a strategy, right? You never get rid of, at least us, we never get rid of any genotype. You never know what could be the hidden strengths, right? But definitely you want to prioritize maybe corals that are doing well at that specific time. Also, going back to the example that we were given before, we had a genotype that was growing slower than other genotypes. And then what we did through this wrapper, we can see where that genotype is replicated, and maybe it's replicated in other nurseries, right? And in other units. And then go and check in that nursery if actually that genotype is also performing in the same way, right? And then you can do it for every species, for every unit, for every genotype. These are very important information when you wanna plan, right? Like pick your genotype ahead of time. Also, a very simple practical use is what we did yesterday, right? We needed to harvest more than 100 corals for the field trip and outplanting. Where are these corals? Where can we harvest this coral to outplant at Oz Lake? Some of you asking, and said, we picked them at the nursery at Buddy's Reef. Are there enough corals at Buddy's Reef, or should we go at knife and harvest this? The same, right? You don't need to go there. We know it because we can just check it on the database. So, very important. Or, as I said before, all the question regarding planning ahead of time in terms of big and larger projects. But our success is measured, as I said yesterday, once we outplant the corals. You can be great at producing, but you need to move the corals on the reef and establish if you're successful there. So again, to establish a monitoring system there, first we ask, okay, what I need to know. I want to know if I can do more with the resources that I have. Can one person outplant more corals, for example? Or are the people trained enough? Why there are some people that can outplant 50 corals in one dive and 10 corals in one dive, or 5? What is happening? Do we need more training? Should we allow only highly trained people to outplant? This is just an example of efficiency, right? Also, assess effectiveness. Once we outplant the corals, are actually sticking there? If they all dying or they are rolling or getting detached, are we actually effective in what? This is a very good, I want to have feedbacks on the technique very fast, not waiting for a year to see if it worked or not. Of course, you measure success in terms of survival and other metrics, right? For example, more related to the ecosystem. What's our impact on the ecosystem, right? Are we actually restoring coral populations here? So how do I measure that? And based on all these indicators, adapt your strategy. So there was no one fits all. So again, to tap into a lot of different protocols. We started for sure, when we started, we were doing a lot of in-water surveys, which I think are very valuable for different reasons, and we will see why. But then I also wanted to know how our sites were developing, right? What was the overall performance of the site? And that's why we also integrated with photogrammetry. As I said before, I want to know what was our impact on the reef. And then We also started doing reef ecosystem assessment. And of course, we needed to know how to track our coral breeding program, right? Fertilization, settlement, and all this metric. So overwhelming. And then I decided, oh my God, we're going to spend more time just collecting data than again, outplanting coral. But it's very important. So again, let's try to keep it simple. So we decided that we were going to monitor our corals underwater just in the short term for the first year. And I will explain you why. First, I extremely value the underwater monitoring because there are things that you can see through pictures, or it takes a lot of time to actually analyze picture, right? So after our planting, we go immediately there to see if there's something to be adjusted. But then we go back to our plantings after 3 months, 6 months, and a year. That's it. And what are the information that we record? First, as we did in the nursery, you need to track your planting. So very basic metadata, the species, the genotype, how many corals. As I said before, the outplanter, not because we want to point the fingers, oh, the corals that Karen outplanted were rolled, right? Not because of that. It was more general, like how much training we need, or is it something happening? Do we see a pattern there, right? That needs to be addressed. Or maybe just go back to Karen. Karen, did you have some difficulties when you outplanted those corals? You know, it was the very surgy, something that we can note. The outplanting techniques, cement, time, wedging, transfer time. We spoke about that yesterday. Where are these coral coming from? Did they have maybe a 30 minutes transfer before getting outplanted, or 20 minutes? So we record that. And some notes, of course. And then with in-water survey, so like I said, after 3 months, 6 months, and a year, we go there. And first we need to check, are the corals still there? For each cluster, right? Do you still count? They say yesterday we outplanted, let's say 5, 6 coral each. In the database we recorded. You come back after 3 months, are the 6 corals still there, right? Or they got dislodged. The percentage of live tissue, very important. And then if there is like recent mortality versus all mortality in the percentage of the dead tissue that you see. And then if there is a recent mortality, we score if there is a disease to understand what's the stressor going on there, right? Predation, competition. The only data that we collect underwater in the long term, of course, is spawning, because you can't do it within only 1 year, right? You need to come back year after year for that. But then time passes, and that's what was happening to us. Like, of course, this is not 1 year, but it's like maybe 2 years. But already after a year, our 10 corals are not 10 corals anymore. They fused, and they were becoming 1 coral. So you go there and you score, and you're like, okay, I don't have 10 corals anymore. I have 1. So the database will read it as like, oh, you lost 9. That's not good. In reality, it's not what happened, right? The corals are still there. If there is a storm, for example, and the coral got dislodged, corals are not there anymore, but they're still at the site. And my goal was to restore the site, in particular for branching coral. Corals are still there. So, but in the in-water individual monitoring, you couldn't capture that. So we moved to photogrammetry. So after a year, we keep every year, and then after 5 years, maybe after 2 years, we change the frequency a little. We monitor our site doing photomosaics. This is actually the site that you were diving yesterday at Oils Lake. This is a photomosaic. It's about 700 square meters, about 1,200 pictures here stitched together. Like I said, with photomosaics, you can zoom in and really see the details, and you can track actually where the corals after storm move, likely, right? Or they're still there. Or even if you don't unable to track, when you measure coral cover, they are still there, right? And they still contribute to the success. So with a specific software, at the moment we're using TechLab, we analyze the photomosaics, and we can extrapolate coral cover, average coral size, the density of the corals at the area, like I said, track dislodgements, the overall restored area, and see how the thicket is expanding or contracting, right? Not like zoom out, eagle eye view of the site. And of course, a picture says more than 1,000 words. So, and I will give you some example, because when you're going to, between us, we can talk about 16% coral cover went down to 5% coral cover. what does it mean for a person that is not working in our field, right? Is it even good, 20% coral cover? It's like, it seems quite lame if you say like that. In reality, it's actually a good coral cover, but it's the reality. Like, and you go and ask funding and you do presentation, you talk to people that don't do reef monitoring, right? So I needed a better tool to show the before and after. So great for data collection, photomosaics, but also for communication and education. This is just to say, like, about the picture, right? This is like, maybe Caroline, I need to give you this for your database, the WWF. I got this picture, and this is actually Carsill, a site at Bonaire in the 80s. And you see how the site looked like, right? And how the population of Elcon coral was thriving there. This picture is very useful when you want to, should I outplant a Carsill more, a crop of Amata? I think just this picture says a lot, right? So this is the site. Oh no, don't tell me that you miss it. Oh, I don't know in Dutch.
It's not the same what I don't consider that I can
I'm sorry. So post a video of the photomosaic panning over the entire site. And just bring it around. Okay. Let's see, because it will look right. You see also our analysis, and we will have the economies of it. So this is a pan on the side, and then you see how we apply all the economies and how for over that calculated automatic. This is the side. You see the outline of all the colonies. The light yellow are the white colonies. The orange are the outplanted cores. So okay. So, and this was January 2023, when we didn't know the Schedule D was going to be recorded in Bonaire just a few months after. So at that time, our interest at that time was only elkhorn coral, right? And these are all the colors I showed you before. This is just a little segment actually of the site. The site extends for more than 1,000 square meters. And these are the orange are the one that we have planted, the light yellow, the wild population. Automatically the software gives us coral cover, as I said, the amount of colonies, the average diameter. But then Schedule D arrived, and we needed to go and look for brain corals as targets of our coral breeding program, right? And I knew that this site, and you can see it in January, was full of brain corals, right? We call it brain coral paradise, because you can see every species of brain corals there. So for us to target it for spawning was like easy work. But then we went there 8 months after Schedule D, and we realized that there were barely colonies still alive. So, quite sad, but it's the reality. But then I remember we did this photomosaic just before. I was like, what a great opportunity to actually measure what was the impact of Schedule D at this site. Because this site goes from 1 meter depth to 5 meters. So it's not monitored with the regular reef survey by the park. So we didn't have any historical information on that population. So this was actually, I think, the only picture that we have of the site before Schedule D. So out of curiosity, with a little bit of spare time, I decided to outline every single brain coral, just so the Diploria species and Diploria labyrintiformis present at site, all the pink outline brain corals, and they were all alive, right? And we're talking about almost 900 colonies. And then we went afterwards and we actually validated underwater this photomosaic, going colony by colony and see which one were dead, infected, but still alive, and healthy. And this was the result. Out of 898 colonies, 689 were dead. 59 also still infected but alive. And we lost about 159. So, sorry, that was the healthy, 159. So we only had barely almost 17% of the colonies alive. Very sad story. I didn't want to bring any very deep negativity here, but I think this is an important application of photomosaics, right? So to consider. And now I'll give you maybe a more positive application, right? So photomosaics are also good as a baseline, right? So this is a site in the south, Point Birken, 500 square meter. We want to restore the stagger population there to start with. So before coral reefs have been outplanted, we did these photomosaics. And then based on that, we started thinking, okay, how many corals we need to outplant there? We set the species, which genotypes are we starting with? Density, like how many corals per square meter we want to place there. And then we use the photomosaic to actually pre-plan where we're going to put our cluster. And this because also was done collaboratively with a PhD student, Ellie Blanchette from Florida State University, because she wanted to actually measure performance of our outplanting. She also the person that did all the genotype tests. So she really wanted to follow this genotype, how were they performing on the planting. So we kind of worked together on make the best plan, also for her in terms of research and for us in terms of outplanting. So we discussed, right, what is good for me in terms of maybe positioning of the coral, what was good for her in terms of replicate across different gradients to maybe answer specific questions, right? And that's how we planned. And then with this plan, we went underwater and started outplanting corals. And then the photomosaics afterward, we did a new photomosaic with all the corals there. Actually, with the picture, this I showed you also yesterday, we were able to tag on the pictures, so without having to use the label on the waters, right? We use it anyway for the in-water survey for the first year, but after the first year, if you use this method, there is no need to keep plastic on the water. So it's very useful for implementation as well. This is the analysis of the site after a planting, actually 1 year after a planting. And then I thought we were done. Okay, in the water surveys, first year, photogrammetry, is it enough? Well, then we couldn't really talk to the park or with the data that they were collecting because they were doing reef surveys. How we integrate our information with what they are doing, right? How our work impacts the overall reef ecosystem. Right, so we didn't want to reinvent the wheel, and we want to have the ability of our results also to talk to theirs. So we adopted the same system, GCRMN, Global Coral Reef Monitoring Network. So we adopted the same protocol. We just started last year, and we're doing with the same frequency of the photomosaic, so we can actually associate information. So we can collect information at our site about fish population, algae, invertebrates, and coral recruits. And and have a more, let's say, overall picture, right, of the site. Also, sometimes happen that maybe the park is not doing the GCRMS specifically at that site, right? So we wanted to know, for example, if, what is the for example, we want to have a reference before our planting, and in some sites we don't have it because we already started, right, our planting there. I said it started last year. So what we did, we started doing also GCRMN to a site that is nearby located and where we didn't outplant it. So we collect data at the outplanting site and just nearby as well, repeating, so we can see if there is a difference, use it as a control, let's say. Summarizing again, I just want to share with you how important it is for us to have these sort of indicators for better planning for larger projects, to be able to strategize. In particular, if you are asking for grants, and for example, most of the time they ask you, okay, how many square meters are we talking about? So you have your area, and this indicator helped me to establish a timeline for the project, to have a better plan on genome and species selection, risk management, again. So if you have a project that is so important to you, do you want to produce all the corals only in one nursery? Or maybe just in case there is a heavy storm or a hurricane, maybe it's better to distribute your production over different nurseries so you're sure that you can produce those corals, right? So have a little bit of risk management as well, informations, and also it helps you to allocate your resources. Again, all this information, like our database, are crucial for the daily schedule, weekly schedule, monthly schedule. Where do we need to work? Where do we need to frag? Where do we need to outplant? Very, very easy. You can just check all the nursery and see, okay, we need to work here and here. Corals are dying at night. Let's go, now I can see what's going on, right? Also, because when the project gets big, sometimes you have different teams working at different nursery, and something that is in the memory of the surveyor at one nursery might, or this surveyor might not talk with the other. So if you don't have a database that actually can reveal there is something going on that is weird, right? If these people, if you have multiple teams, again, they are working in separately, sometimes you might not grasp there is something going on simultaneously, same thing at multiple nursery, multiple sites. Reporting never been easier because you don't need to analyze data, they're already there. So I'll give you an example. We were running a project together with the park, funded by the Resembit in Lac Bay, and they were the project leaders. So I just needed to report on a trimester basis the performance of the nursery. I just send a screenshot of the nursery of the database every 3 months because they have mortality, the corals produced, how the corals were growing, and all this information. So I didn't have to do any analysis. And that's it. So thank you so much for your attention. I want to thank you, second Kedrick Willow. That's the genius behind the database. He's a volunteer database developer that took the time to really learn what we do and understand our lingo to translate it to her lingo and create this, we call it the Ferrari, for how it runs. And also I want to thank, of course, all our founders, sponsors, supporters, and partners, and of course DCNA for allowing us to share part of our work. Thank you. Oh yes, of course. I always
any questions?
share away for questions. Yeah.
The data that you collect when you outplant, do you also collect that for the training outplants?
No, exactly. That's a very good question, right? We can't monitor everything. So training was very hard, the training side, because they keep adding corals. So for this, but of course, the training sites still contribute to our overall aerial restoration, like the overall area. So we do it sometimes with drones or with photomosaics, but we don't do in-water surveys for this because it's very hard to track.
I guess the mosaic, do you, as it's quite shallow, you were saying percent size, it's only down to 5 meters. Do you do it with like divers, hooters, and cameras, or you do it totally behind the boat with cameras?
So for a long time was only one diver with one camera going back and forth, hue pattern. And now, because the sites are getting quite large, we mount our camera on a DPV, so an underwater scooter that allow us to be more precise and cover wider area more efficiently as well. So we actually doubling the size of the site. We didn't double the amount of picture actually, because probably we became much more efficient in the way we're doing new pattern, more consistent because the DPV allow you to keep a very good pace. So actually we have been much more successful in our photomosaic since we started adopting that. But for small site, I mean like 500 square meter, one diver can cover in one dive very easily.
I hear you present, I'm just amazed at your what you've accomplished and your persistence and what you've developed. So, question is, this database, this analysis tool, is it open access? Can anybody use it if they follow your protocol?
Well, the database itself is an access database that was developed by this person, so that maybe is willing to help others to develop. I think each database is tailored for the project. So I just want to share with you, I think you should start more than adopting a database, more thinking about, okay, what are the information that I want to know? And instead of having thousands of Excel sheets, for example, getting help for somebody that will help us to have this information properly collected, more safely secured, also the data, and have reports in automatic. So you don't need constantly going and open your outplanting database, your nursery database, your all this database and collect this info and try to extrapolate this information. So it's more about what I need to know and start from small. This didn't happen. This wasn't as big as it is now, you know, and as well with your production or outplanting or photomosaics as well. Start small, and then it's much easier to be, you know, successful when you start small and you start increasing afterward, right? Think always that you will get bigger, of course, also when you think about your database, but start with small. Don't try to be too ambitious in your data collection.
Thank you so much, Francesca.